We really we do these kind of things because we want to emphasize how important participation in something like UK Biobank is, and not just participation in the first place. You know, I mean, just turning up for the assessment centre, 2008, I think Andrew said. You know, that has already provided an amazing, unique, groundbreaking resource. But also the fact that, you know, a lot of you, I mean, are so eager to kind of constantly continue contributing data over time, and it is the combination of that engagement which really does make this such an important study for scientists, not just in the UK but around the world. And we really can't demonstrate that in the course of 2 hours, but I think, you know, we really want to try and get that across, especially as scientists. So just to kind of give you an insight into why UK Biobank is especially important study. So UK Biobank is set up to investigate a wide range of things. In terms of health outcomes, things like common exposure, so take for instance smoking or alcohol, with big effects like cancer. These are now, you know, these are fairly well known. But the causes of most disease and ill health are a combination of things: lifestyle, environment, physical condition, genetics. You know, we know a lot of the common things that, you know, you need to do to modify, but in order to get insight into a lot of disease and ill health, we need to have a lot of data. We need to collect a lot of data on the same people so we can explore how these different things interact. But not just how they interact, but things that are maybe less common exposures. For that, then we need a lot of people, because small effects from common exposures or large effects from rare exposures are difficult to detect. You know, we can see these associations with something like smoking and cancer on maybe, you know, a few thousand people, but if you really want to see what is causing a lot of ill health and disease, you need to have large studies, hence 500,000. But also, and this can't be stated enough, it's complicated. I mean, it is really complicated trying to tease out and disentangle like all these different things that are going on. You probably get this yourself when you see the newspaper and one day it's saying one thing, one day it's saying another thing, and you think, what's going on? Why can't they make up their minds? But it's because it is complicated, and often this message isn't really getting through. So we need to collaborate as scientists, you know? We need to work together with experts across the world so we can really make the most of the data we have. So this is where UK Biobank comes in, because a lot of studies have these elements individually. You have large studies, you have studies that are a bit smaller, but they have a lot of data on things, but then those studies can't, you know, investigate the cause of disease that are fairly rare. And then you have studies that are usually not necessarily that accessible. You have to collaborate with the people who've designed the studies. We don't do that. We're open access. We're open access to scientists in the UK, scientists worldwide. So we have a combination of a lot of data, so the data collected at the start, but also ongoing data collection. Large, 500,000 people, but also that it's accessible to scientists worldwide. So I'm just going to go back over the whole kind of recruitment assessment again, just to kind of you know, remind you of how UK Biobank was set up and designed. So between 2006 and 2010, a mailed invite was sent to those aged 40 to 69 years old. So this age range is, you know, chosen as a particular age range because a lot of diseases, you know, and ill health are now being caused around about middle age or later on in life, especially living in, you know, westernized countries. So this is the age range where we're now going to be investigating a lot of these diseases and conditions which are much, much more common. So everyone is registered with the NHS and live within 25 miles of one of 22 assessment centres. So these assessment centres were distributed throughout the UK, England, Scotland and Wales. So in order to get 500,000 people, we had to just send invites to a lot of people, to get that kind of response rate. So we invited more than 9 million people. So you're a very select group, for actually taking part in this study. So 5% response rate. And that's the number we were achieving, because the study was set up to achieve 500,000. So we had just over 500,000 participants. And Newcastle was actually, with 37,000 participants, this is the area with some of the most participants in UK Biobank. So when you all came to the assessment centre, you went through this long process where, as I said, we wanted to collect a lot of data. So to do this, you had to go through quite a bit. So at the beginning, you would have had a touchscreen questionnaire. This collected a range of information on things like demographics, lifestyle, diet, and early life exposures, family history, but then also things a bit more focused, like hearing test, because hearing, age-related hearing decline is a very common condition which we don't necessarily know that much about. Cognitive tests as well. And then a verbal interview. So then the verbal interview was used to get a lot more detail on things like medical conditions, specific medications, and operations. And then a whole range of physical measures were collected. Blood pressure, hand grip, BMI, but not just BMI, a range of anthropometric measures, a range of like waist to hip ratio, waist circumference. Spirometry, so spirometry measures lung function. A heel ultrasound, which measures bone mineral density. And then I'm not too sure if this was performed in Newcastle, but some eye tests were also rolled out at the end of the study. So I'm getting some one nod. So I'll take it that they may have been used in Newcastle. So then this was to capture, eye conditions and eye functioning, and also an exercise ECG. And then some blood samples were taken, as well as urine samples and saliva samples. So, a lot of data was collected at recruitment. So we have all this data on 500,000 people. So, great, okay, we've got this, but then it's what do we do with this data? So from the samples, from 500,000 samples, these have all been split up into 15 million aliquots. So that means that for each participant, there are multiple samples, or, it's all split up. And they're stored in this big freezer, which has this custom designed robot arm, which goes back and forth. I've never had a chance to play with it, but it does look very exciting. And it was designed especially because having this many samples on this many people meant that you can't literally go into this freezer and pluck out a sample and start performing measurements on that sample. This robot arm, you can type in which participants you are looking for, and it kind of zooms around and picks stuff up, and it is very technologically impressive. So what we've done with the samples is we've measured the genetics on all participants. So this is then the largest database of genetics available in the world. And I mean, I'm sure everyone knows this, is that things that causes disease or ill health, or kind of, actually let's flip it another way, things that causes healthy aging as well, are a combination of lifestyle, a combination of environmental exposures, but then we also know there's a genetic contribution as well. But it's not as simple as it is often portrayed. We kind of maybe think of it as, your genes are your destiny, or you've got this particular gene and then you're going to get this. But it doesn't actually work like that. So your genetics contain a lot of information, and it's a small part of that distinguishes us. And a lot of us share, most of our DNA. So what we measured are these small bits of the DNA that account for that variation between us, and these are called single nucleotide polymorphisms, or SNPs. So SNPs are variation in DNA. So I've really kind of sort of simplified this, but let's say you have two people and you have their genetic code. So you have the first one on the top and the second one below, and you can see actually they have 4 different letters, A, T, C, or G, and most if you line them next to each other, then they share a lot in common. But then, so this one on the top has C, this one on the bottom has G, so that's that bit of variation in the genome. So this person then has blue eyes, and this person has brown eyes. And I've put this note because what I've said is essentially wrong because eye color is very complicated and it doesn't work like one gene, as I was saying, but how these kind of tiny little changes here and how they occur throughout the genome, these contribute to different things like, say, eye color, but then also things like maybe having slightly higher cholesterol, slightly lower blood pressure, slightly increased risk of having dementia. So, being able to have this on 500,000 participants is a really, important resource to be able to look at the contribution of genetics to disease. So we've measured 820,000 of these. Exome and whole genome sequencing are currently underway. So exome sequencing, that measures the part of the DNA that is codes for different proteins, so is responsible for, so it's basically capturing more than what we've already measured. And then whole genome sequencing, that just measures absolutely everything. So these are really groundbreaking. The problem with this is that there's so much data that we need to have like really dedicated computer resources because the amount of data that this produces is so huge. But also from the samples, we can measure biomarkers as well. So a biomarker is, just to simplify, a biological indicator of a certain process. So for example, cholesterol levels are a biomarker. They can measure your cholesterol levels, and then that can be an indicator of whether you've got cardiovascular condition. So we've also measured a range of these as well. These can give us a lot of information about primarily, I would say, risk factors for disease. So finding new biomarkers that could increase your risk of certain diseases. And we're also in the process of looking at whether from these samples we can measure infectious agents as well. So we've done this already in 10,000 samples. We haven't received funding to do this, but basically this would be this would be a unique thing to look at, especially in the context of this kind of study, because infectious disease is not often measured in a study of UK Biobank. We know that infectious diseases such as HPV can cause cancer. We know that a range of infectious diseases can cause a variety of different kinds of cancers. So, but there's not much data where we're able to look at all these different kinds of infectious diseases with a lot of different kinds of data. For example, risk of cardiovascular disease, where there's some suggestive evidence that infectious diseases have a role in cardiovascular disease, but this just has not been measured in studies where you're able to look at this in any kind of detail. So even having this for 10,000 participants is unique. So there's even some interesting ones like, EBV, Epstein-Barr virus, so this causes glandular fever. We know what a lot of these infectious diseases do. A lot of them are actually asymptomatic, and you can see they're very highly prevalent. So 95% of people have Epstein-Barr virus, have been exposed to it at some point in their life. But there's been some evidence that this is linked with multiple sclerosis. So, 95% of us don't have to worry about that because it's going to have a very small effect if it is, but you can't look at that in a lot of study designs. You could only look at this in a study designed like UK Biobank. So I mentioned kind of like the data we collected at baseline, the assessment centre where you turned up in 2008. I've mentioned about the kind of ongoing work, so measuring the genetics, measuring the biomarkers as well. But a really important part of UK Biobank is the fact we have this ongoing engagement with you as participants. The fact that we have this additional data collection so that we're trying to capture a range of information on things that we weren't able to get a baseline at the assessment centre because you were there for a couple of hours. People wanted you there for like 6, 7, 8 hours because there was so much information that we wanted to collect. But obviously that's completely unfeasible. So we've got other ways of keeping in contact and trying to get more information on certain things. So online questionnaires are a really useful way of doing this. We have email addresses, I hope, for pretty much everyone in this room, but for about 2/3 of the participants, so about 350,000. And every 6 to 12 months, we send one of these online questionnaires, and they're really useful for getting in-depth detail on certain things that we weren't able to collect at the assessment centre, but then also that you can only really get through a detailed questionnaire where you really go into detail about certain things that you can self-report. So in 2011 and 12, we did a 24-hour dietary recall questionnaire. So this was basically asking what did you eat in the previous 24 hours. This was ran 4 times to capture seasonal variability because people often eat and change their diet at different times of the year. 2014 and 15, we had a cognitive function questionnaire. So this is really useful because we did cognitive function at the initial assessment centre. By having it in the online questionnaire, then we're able to see change in cognition over time. So for example, declining. Occupational history. Again, we're not just focusing on biological stuff, genetics, and the biomarkers I mentioned. We're interested in lifetime exposure to things like occupational history, things that you are exposed to in particular occupations. Mental health. A lot of this can be things that we just can't capture any other way unless we actually ask people, on how they feel or what kind of mental health issues they've had. Digestive health, and then these are some of the questions that we have designed for the next few years. So pain, I think this is actually already started, so I don't know if you would have received anything yet, but then this is asking specific questions on different kinds of pain. So, it's really detailed. You're able to get what parts of your body, you might have pain, how long that pain has lasted for, how severe that pain is. Because all these different kind of aspects of it, obviously make a difference than simply saying, do you experience pain now and then? Food preferences. So, what kind of food people prefer, not so much what you eat or drink, but what kind of, comparing, do you like bitter food, do you like sweet food, preferences to certain kinds of food. Sleep, so sort of sleep quality, and also quality of life. And I think some of these things are really important because they can often be overlooked in these kind of studies. These might seem somewhat subjective, but they're not. They're actually important. When people report their own quality of life, that is an important measure that we want to try and understand what contributes to that. I don't know if anyone here has worn one of these. It's an accelerometer. Okay, yeah. Did you? Okay, I thought you put your hand up, but I wasn't. So this is an objective measure of physical activity. So it's just a sort of a wrist watch, but it kind of looks like a wristwatch. And what this does is capture your physical activity as you're just moving around. And this is quite, this is clearly quite useful because we ask questions on physical activity, and probably won't surprise you, but I think like 95% of people vastly overestimate the amount of physical activity that they do. So having an objective measure is really useful, but not just to get an idea of the amount of physical activity, but also patterns. What sort of physical activity people are doing over time, the intensity of physical activity, so periods where people aren't doing so much, what kind of time they are doing physical activity. So this was collected for 100,000 participants. They wore these for 7 days, and we've just finished this where it's repeated in 2,500 participants 4 times over 1 year. So again, having those repeat measures is really important. It's really informative. It kind of tells you how consistent people do things, or what sort of changes over time. But you can actually pick out like really interesting patterns from these data as well. So I showed you this is what it looks like. This is what the output looks like, which doesn't really seem to tell you too much. But when you compare people, you can see like on this dashed line, that's an evening person. You can see on the solid line, this is a morning person. So you can pick out patterns of sleep just from something like wearing a monitor on the wrist. And then this one is someone who cycles to work, because their peak here around about commuting time, drop off here, where I don't know what this person doesn't seem to do too much at all actually, but then this one is at work and then doing an office job or something, and then this is when they cycle back home. But a lot of this work has been done because we have this kind of scale of data, and we're able to extract really novel and interesting and unique things. And something like sleep, that might look kind of, okay, that's interesting, well, what's that telling us? But sleep quality is a very important contributor to health. So I won't go into any detail about this because we have 2 talks specifically on this. But just to briefly say, we're now undergoing a really major reassessment where we have 100,000 participants that are coming in to do this multimodal imaging assessment. But as I said, we'll talk about that a bit more later. So those are the ways of following up participants, as I said, following you up. So online questionnaires, these are all active things. You have to actively be involved to take part in these things. But even if participants aren't active, we still are gaining a lot of useful data through passive follow-up. So we do this through linking to medical records. So as I said, the recruitment was based on being registered with the NHS, which is really useful in the UK because we have this National Health Service, obviously, and like everyone is basically registered with it. Everyone's data is in these medical records that we are then able to link back into UK Biobank. So currently we have death registry data, so death, which also includes cause of death, cancer, so types of cancer. Also we have different kinds of cancer data where it's like severity of cancer, how much your cancer has spread, that kind of stuff. And also hospital inpatient records, so this is when you're admitted to hospital, so diagnosis for that. But we will also have primary care data soon, and this is going to be very, important and useful source of data, because most of our health ailments, we go into primary care, we go and see a GP. We have things like blood pressure taken, that kind of stuff. We don't go to hospital every time we have like a bit of pain or something. So this data, primary care, hasn't been linked into a study of this size before. So having this really does open up very unique possibility for researchers. Andrew, will you stand up because I completely overran? So okay, well I'll skip over these, but these kind of give you an idea that the numbers will be fairly substantial. Not because you're unhealthy, actually you're complete opposite. You're much healthier than people who didn't participate, but just purely because of the number of participants that we have, 500,000. But I think what you can see is that we're focusing on these are a range of kind of conditions, things from cancers, diabetes, heart attack, but then things like hip fracture. Hip fracture is a really large cause of ill health and and can impact quality of life quite substantially. So just to kind of quickly summarize, the research interest in UK Biobank, since we made the data available in 2012, is absolutely huge, but it's increasing all the time. We have now about, slightly out of date, so this is about 1,700 different research groups working on specific projects, specific applications. And these are spread, all throughout the world. And one thing to highlight is that a lot of these are collaborating, so there's a lot of cross-collaboration worldwide. A lot of publications, and yeah, I was going to just kind of quickly summarize some examples of some of the publications that have come out. But I think just to focus on this one here used both the genetic data and it used the lifestyle data. So you could only do this with having all that information. And it looked at the benefit of having a healthy lifestyle and the risk of having a stroke. And what it did was kind of derive this healthy lifestyle score. So if you did 1, 2, 3, or 4 healthy things, then you had a score of 4. If you did 2 of those, you scored 2, so on, so on. And then it split the participants into genetic risk of stroke, so just into like thirds. So top third is the highest risk, bottom third is the lowest risk, etc. And then looked at how much of an impact healthy lifestyle had on risk of stroke within those who had a high genetic risk, a low and intermediary. And it found that, unsurprisingly, if you've got higher risk, genetic risk, then you're more likely to have a stroke. But those who followed a healthy lifestyle had made a massive contribution to their risk, despite the fact that they were in the high genetic risk. And is almost comparable to those who follow like an intermediary kind of lifestyle in the low genetic risk. So I think in a way it's kind of common sense, because we try and get across the importance that genes aren't destiny and it's not as simple as nature or nurture, but actually it's a combination of both. And even in this age of a lot of genetic testing or whatever, having a healthy lifestyle or other environmental things can have a massive impact on your risk, even if you have a high genetic risk in the first place. So thank you for listening.